Hello everybody, this is Aloysius Guy in a new tutorial, and in today's tutorial we're going to understand the different roles and function of the different RNA types that we have in our cells. So stick to the end of this video. Please, before we start this tutorial, please don't forget to go to the description so you can find all the chapters and to go quickly to the chapter that you're interested in. So in our last video we showed how RNAs are synthesized. If you want to check that, you can just click on the link that will appear here. Okay. And just to make a quick recap, so we have different types of RNAs. So we have messenger RNA, transfer RNA, small interfering RNA, microRNA, and ribosomal RNA. And every one of them has a different function. And we also said that mRNA is the only coding RNA, which means that it's the only RNA that is imported for, or that is translated into proteins, okay, and codes for proteins. The others only assist in the either assist the production of proteins, or they just have different roles. The main role of these 5 different RNA types are in the cytoplasm. So they are all synthesized in the nucleus, but their main function is in the cytoplasm. And then we have the other 3 types, which are small nucleolar RNA, long non-coding RNA, and the small nuclear RNA. And also they have different roles. However, they are also made in the, although they are also made in the nucleus, their main function it's within the nucleus, okay, and that's the main difference between these types. So once an RNA is made within the nucleus, as we can see here, so this is the nucleus, okay, and this is the nucleolar, okay. So within the nucleus wall, you will find these gaps. They are called the nuclear pores, okay. So the main importance of these nuclear pores is that they allow the movement of, for example, nuclear RNA exports. For example, like we said, the 5 different RNAs that main function is in the cytoplasm. So they can move out of the nucleus through these pores. Okay, so some other RNAs and also proteins, they might, for example, the small nucleolar RNAs and long non-coding RNAs, so they might go to the cytoplasm just to get processed, and then they go back to the nucleus. So that's the way that this happens. Also, there are some nuclear proteins that are made outside of the nucleus, but then they need to get to the nucleus. So all these movements it occur through what is called a nuclear pore, and that's very important to understand, because as we said, these type of RNAs, their main function is in the cytoplasm, and that's the way that RNA is exported. So now we will start with understanding the role of the first mRNA, the first RNA type, which is messenger RNA. Okay, so first of all, so the messenger RNA is involved in a process called translation. Okay, so translation is the process of synthesizing proteins. We will cover this topic in our next video, but here we will just show very generally the role of mRNA within the cytoplasm. So this is the nucleus. Once the mRNA is synthesized in the nucleus and then moves through the nuclear pore into the cytoplasm, we have this mRNA now in the cytoplasm. And as we said in the previous video, that these modifications in the mRNA are very essential for its role in translation. Okay. So once mRNA is in the cytoplasm, they will interact with a protein, a complex protein called ribosome. Okay, so ribosome is made up of 2 subunits, a large subunit and a small subunit. Okay, so what happens is that the mRNA will serve as a template for the production of protein. Okay, so this peptide chain is being made through the ribosome. Okay. So as we can see here, the ribosome will identify the mRNA and start moving through the mRNA. Okay. The order of the order of nucleotides within the mRNA will determine the order of amino acids in the polypeptide chain. Okay. Therefore, the order of amino acids will ultimately determine the structure and the function of the synthesized protein. So this process is very essential for producing all the different proteins that a cell might have, okay? And the movement of the ribosome is from the 5' to the 3', Okay, so they bind here close to the 5G cap and then go through toward the poly A tail. Okay. So now let's have a look at the other RNA type, which is transfer RNA or tRNA. Okay. So the structure of the tRNA is a bit more complex than the mRNA, as we can see here. Okay, so once it's transcribed in the nucleus, it will produce a precursor tRNA. Okay, so which will undergo specific processing like splicing and also some modification to its nucleotide to produce a mature tRNA. The mature tRNA will move out of the nucleus into the cytoplasm. Okay, so once this tRNA is in the cytoplasm, it will bind to an enzyme called amino acid tRNA synthetases. Okay, and the main role of these amino acids is to bind. The main role of this enzyme is to bind a corresponding amino acid. Okay, so every amino acid will bind to a specific tRNA. So this specificity occurs because of this specific sequence that is carried on the tRNA. So as you can see in this area of the tRNA, we will have 3 bases. Okay, so every 3 bases are referred to as a codon. Okay, so every codon corresponds to a specific amino acid. So for example, AUA corresponds to an amino acid called isoleucine. Okay, and so on. So, and also this codon will be complementary to a codon on the mRNA. However, we will get into more details about that process in the next video when we discuss the translation process. Okay, so that's the first step that happens. Since there are 20 different amino acids in the human body, there are 20 different synthetases, one corresponding to every amino acid, and this also governs the specificity of binding specific tRNAs to specific amino acids. The second step involves using ATP or using energy in the form of ATP. Okay, and the reason the energy is needed is to form a covalent bond. Okay, so a covalent bond will be formed between the amino acid and the corresponding tRNA. Okay, so once this bond forms, the amino acid, the tRNA will dissociate from the synthetase. Okay, and this tRNA is now called a charged tRNA, which is a tRNA bound to its corresponding amino acid. And that's the main process of how tRNA is bound to amino acids. However, this is not the end of the role of tRNA. They will next go and bind to ribosomes and get involved in the translation process. However, we will discuss this in the next video, so stay tuned for that. So to give you a very quick glance at the how the tRNA binds the ribosomes. So here we have an mRNA. Okay, so once the mRNA is produced in the nucleus and then moves to the cytoplasm, they bind the ribosome, the small units of the ribosome. Okay, and then the ribosomes will start moving along the mRNA, and as we can see here, each three bases are called a codon. So this is one codon, this is a second codon, and these codons are the codons that are identified by the tRNA. So the process starts with, as we already know now that the subunit is made up of, the ribosome is made up of 2 subunits, the small subunits which binds the mRNA and the large subunits which bind the tRNA. So here we have a tRNA, this is the anticodon on the tRNA, and it bound to its complementary codon on the mRNA, okay? And this is a growing polypeptide chain. So what happens is when that tRNA identifies this codon, a new tRNA will come in place as well. So now there is 2 tRNAs within the large subunit of the ribosome. And what happens is that this polypeptide chain will be transferred to the new tRNA. So, and what happens then to this tRNA is that it will leave the large subunit of the ribosome. Okay, and that's very quickly the whole process of binding tRNAs to the ribosomes, and we will go into much more details in the next video when we make the translation, when we talk about the translation of proteins. So now let's look at a different type of RNA, which is the ribosomal RNA, and we will understand how ribosomal RNAs are also contributing to the protein synthesis process through interacting with ribosomes. Okay, so within the nucleus we have a nucleolus. Okay, and within nucleolus is the place where ribosomal RNAs are synthesized. Okay, so they are synthesized as pre-rRNA, and then they get processed to produce the mature ribosomal RNAs. So the nucleolus has a high concentration of ribosomal RNAs. Okay, so once we have this mature ribosomal RNAs, they will interact with the two ribosome subunits. However, in this case, these subunits will not be attached together within the nucleolus. They will be like separated. Okay. So the large subunit will have two different type of rRNAs incorporated within the subunit, and the small subunit will have a single rRNA type incorporated within the ribosome. So that's what they look like. Okay. So after the rRNAs are incorporated into the ribosomes will move out of the nucleolus and then out of the nucleus through nuclear pores into the cytoplasm. And then the two subunits will join together, as we saw when this small subunit bind the mRNA, and then the large one will bind the tRNAs, and then the protein synthesis process starts. Okay, so which we already saw a little bit about it. So you need also to remember that this incorporation of rRNA within the subunits is very important, as it provides ribosomes structural integrity, and also is very important for their catalytic activity. So the other type of RNA that we're going to look at now is the microRNA, okay, which is mainly involved in silencing gene expression. Okay, so let's have a look at its production and its role. So first of all, microRNAs are produced within the nucleus, okay? And within the nucleus, once they are transcribed, they are called at the beginning primary microRNAs. So primary microRNAs interact with a complex, a multi protein complex. It's called the microprocessor complex, and this complex results in processing and modifying the primary microRNA, making it a precursor mRNA. Once precursor pre-microRNAs are produced, they will move out of the nucleus, okay, into the cytoplasm. So once pre-microRNAs are in the cytoplasm, they bind to an enzyme called dicer. Okay, so the main function of dicer is to further process the pre-microRNAs, producing mature microRNAs. Okay. So as we can see here is that once the pre-microRNAs are processed, a single strand of the microRNA duplex will be loaded on a protein within a complex called the RNA-induced silencing complex. So this complex, the main function of that complex is to bind the microRNAs. Also, it binds an mRNA that is targeted by the microRNA strand. Okay, so once the microRNA bind to the mRNA within the REST complex, what happens is that the mRNA is going to be degraded. Okay, and mRNA degradation, it means that the mRNA will not bind to the ribosome, and therefore the protein will not be sensitized. Okay, and that's how the microRNA works as a gene silencing RNA. The other type of RNA that is also involved in silencing gene expression is small interfering RNAs, or the siRNAs. Okay, although the source of the siRNA is different from microRNA, however, the process is very similar. Okay, so as we can see here, the source of siRNA, it's rather than being transcribed in the nucleus as microRNAs, their source is usually different. So the source of siRNA is either endogenous double-stranded RNAs produced by the cell, or exogenous double-stranded RNAs that came from outside, for example, from an infection, a viral infection. Once the DNA of the viral is inside, that's considered an exogenous double-stranded RNA. So what happens is that once these double-stranded RNA in the cytoplasm, they're going to be recognized by the enzyme dicer. So what dicer will do is that it will break the double-stranded RNAs and then cleave them into small fragments. And similar way to what happens with microRNAs is that the rest complex will bind the mRNA that is produced by the nucleus, and also these cleaved siRNAs will be loaded into the rest complex, and they will specifically bind to their complementary mRNA. This will induce mRNA cleavage and then breaking the mRNA, and then ultimately resulting in gene silencing. So it is a very similar process to the microRNAs. Okay, and these two RNAs are very important in regulating the level of gene expression within the cell. So with that, now we understood the roles of the different RNAs, that their function is mainly within the cytoplasm. So how about the other three RNA types, which is the small nuclear RNA, small nucleolar RNA, and the long non-coding RNA? So our understanding of these RNAs is still evolving. Okay, so their function is not as straightforward as the previous RNAs. However, we will cover their main roles that we know so far. Please don't forget to like, comment, subscribe, and also hit the notification button if you like the video. And let's continue. So the first type of RNA that we're going to cover is the small nuclear RNA. Okay, so this RNA is very important as a component of what is known as a spliceosome, so which is a complex of proteins that is important for the splicing process, which is the process of removing introns from pre-mRNA. So as we can see here, we have the pre-mRNA, which has both exons and introns. Okay, and then here we have the spliceosome. So what happens is that the small nuclear RNAs, once they are transcribed in the nucleus, they will interact with the spliceosome, okay, and snRNAs with the spliceosome together, they are called snRNP. Okay, so this complex will interact with the intron region of the pre-mRNA, results in excising or removing the intron from the mRNA, producing a mature mRNA with only exons. Okay, so the take-home message for this RNA is that it's mainly important as a component of the spliceosome complex within the nucleus. The other type of RNAs, which is the small nucleolar RNA. Okay, and this RNA is important for the post-transcriptional modification of ribosomal RNA. Okay, so just bear in mind that the small nuclear RNA, snRNAs, are important for mRNA processing, and snoRNA are important for post modifications of the ribosomal RNA. Okay, so what happens first is that the small nuclear RNA will be produced within the nucleus, and then they will interact with core proteins within the nucleus, producing a complex called snoRNPs. Okay, so snoRNPs will interact with precursor ribosomal RNAs, and this will result in producing a mature ribosomal RNA that is very important for, as we said before, the structural of ribosomes. Okay, when it comes to the type of processing that the snoRNPs and the snoRNA does to the pre-ribosomal RNA, there are two main mechanisms and regulations of ribosomal RNAs. So the first thing that the snoRNAs does to ribosomal RNA is what is known as pseudo-uridylation, okay, of ribosomal RNA. The second one is methylation of ribosomal RNA. So what is pseudo-uridylation of ribosomal RNAs? So what it happens is that the small nucleolar RNA will catalyze a reaction that converts uridine within the ribosomal RNA to pseudo-uridine. Okay. The methylation is induced by the snoRNAs is that snoRNAs will induce a transfer of methyl group to the hydroxyl group within the ribose sugar of the ribosomal RNA. Okay, these two processes are two of the main mechanisms that these RNAs are affecting ribosomal RNAs, and that's why they are indirectly also involved in important for protein production, because they are important for producing ribosomal RNAs. So now we will cover the last type of RNA in this tutorial, which is the long non-coding RNA, or the lncRNA. And this RNA is mainly involved in regulating gene expression. So how does it do that? So the link RNA has a complex structure. It's produced or transcribed within the nucleus, and also their main function is within the nucleus, and it affects the gene expression in three main ways. Okay, the first way is through interaction with chromatin. Okay, so once the link RNA interacts with the chromatin, they can induce chromatin, altering their conformational or structural conformation. They might also recruit transcription factors. They also interact with chromatin modifying complexes, and based on these changes in the chromatin, the link RNA can enhance or even repress the transcription of a gene. So it can make a specific gene increases in expression, or they can inhibit their expression, and all that depends on what type of modification or interaction they would have with the chromatin. The second thing is that the link RNA can also interact with the DNA, so they can generate a hybrid structure with the DNA, and this hybrid structure with the DNA will result in influencing chromatin accessibility, and this ultimately will affect gene expression as well. The third thing is the mRNA. Okay, so link RNA can also bind to messenger RNA, and this binding will affect the abundance of the mRNA, the stability, the translation into proteins, and also its localization. Okay, that's why the link protein can strongly affect the rate of gene expressions. Okay, however, our understanding of this RNA is still evolving, and these are just the main effects that are known at the moment. And as usual, here are some questions to test your understanding of today's topics about the different types of RNA. And with that, we concluded our today's tutorial. I really hope that you find it easy to follow and understand. Please don't forget to like, comment below, subscribe, and push the notification button so you can get notified when I release the new video. Also, write in the comments any suggestion of any topic so I can make a video on them. And please also, if you want to, if you are interested in just looking at specific chapters, you can go to the description and you can find the time codes there. Thank you for watching.